Good day, this is the Professor, drwillphd, PhD, and today's Friday, November 18th, 2022. It is 6:46 and 36 seconds, a.m. Eastern Standard Time, and today's research, tumor mechanisms. So let's hop to it here. The study finds common, targetable mechanism which tumors use to suppress immune responses. Date November 17, 2022. Source: Ludwig Institute for Cancer Research. A new study has revealed a single protein expressed at high levels by cancer cells across a broad range of malignancies that affects a multifaceted barrier to anti-cancer immune responses in mouse models of cancer, and so shields tumors from immune detection and destruction. The study also describes a signature of gene expression induced by the protein named FMRP that encompasses 156 distinct genes and predicts poor patient survival across multiple types of cancer. The findings and further development inform the selection of patients likely to benefit from immunotherapies and the development of new therapies of this kind for multiple types of cancer. The study has detailed a previously unknown and apparently common mechanism by which malignant cells shut down anti-cancer immune responses. They have shown that the hyperexpression of FMRP, which they and others have previously linked to tumor progression, doesn't directly drive cancer cell proliferation and tumor growth. Rather, it supports the ability of malignant cells to manipulate the types and functional states of immune cells around them in a manner that very effectively subverts immune attack. A protein primarily expressed in neurons, FMRP, has been extensively studied as a factor whose loss of expression during embryogenesis is associated with a neurodevelopment disorder, Fragile X syndrome, which causes severe intellectual disability. The functionality, FMRP, is known to help stabilize the messenger RNA readouts of genes in cells and to regulate translation of that information into proteins, but its role in cancer progression was less clear. The researchers began by showing that FMRP levels are elevated across multiple types of tumors. To examine its function in cancer, they applied CRISPR-Cas9 gene editing to delete FMR1, the gene that encodes FMRP in most cancer cell lines. They then used the engineered cell lines to generate mouse models of pancreatic colon, melanoma, and breast tumors, and compared them to matching tumors that retained their FMR1 genes using mice that either had or lacked intact immune systems. While all tumors grew similarly in culture in immunodeficient mice, the ones that lacked the FMR1 gene were severely impaired in mice with competent immune systems. They were also heavily infiltrated with helper and cytotoxic T cells, which play a central role in anti-cancer immunity. Those with intact FMR1 genes, on the other hand, progressed aggressively and were by comparison so-called immune deserters. Devoid of anti-tumor T cells, when T cells were removed from the FMR1 deficient tumors, they resume growth, suggesting that the FMRP supports tumor progression through its effects of the immune response. The researchers discovered that the gene expression program regulated by FMRP and cancer cells activates multiple mechanisms of defense that supports immune evasion. Among these is a release of factors that variously promote the induction of regulatory T cells, which suppress the activity of cytotoxic T cells or reprogram immune cells, known as macrophages, into a functional state in which they support the growth and survival of cancer cells instead of their destruction, largely by pacifying T cells. The loss of FMRP in cancer cells, meanwhile, not only reversed the immune suppressive effects, but also induce their secretion of a factor that attracts T cells. Additionally, The FMRP-deficient cancer cells release signals that instructed tumor-infiltrating macrophages to adopt a stimulatory program that helped recruit and activate tumor-killing T cells. While FMRP expression itself isn't a reliable prognostic biomarker for cancer outcomes, the researchers reported a signature of gene expression reflecting the regulatory network it induces consistently predicts relatively poor odds of survival in multiple types of cancer. Researchers are hopeful that these discoveries can be translated into diagnostics and therapies that benefit the cancer patients as the hallmark capability of cancers to circumvent immune responses underlies the resistance of many tumor types to immunotherapy. And this is a professor of microbiology, bioinformatics, and pathophysiology. This video was created for educational purposes only. Hope you enjoyed listening. Until next time, have a great day.